Hello, my name is Simran Dua and I am doing the Kras protein. So the Kras protein is located on the inner leaflet plasma membrane. This is part of the RAS-MAPK signaling pathway, which drives many cancers such as pancreatic, lung, and colorectal. It is ubiquitously expressed in epithelial cells, hematopoietic cells, neuronal cells, endothelial cells, and fibroblasts. It normally functions by transmitting extracellular signals to intracellular pathways, controlling key cellular processes. It promotes cell proliferation through controlled cell division and prevents premature apoptosis. It also allows for cell differentiation, which helps stem cells specialize into different cell types and mature into functional cells. So Kras is a small GTPase, meaning it functions as a molecular switch, cycling between an active GTP-bound state and an inactive GDP-bound state. Kras has a globular shape with distinct surface loops and helices that play roles in its interactions with other proteins. It has a G domain, switch 1 and 2 region, P loop, and a hypervariable region. Kras is a monomeric protein, so it does not naturally form subunits like a dimeric or tetrameric protein. However, it does interact with regulatory proteins like GEFs and GAPs. Kras has a primary, secondary, and tertiary structure, but it does not naturally form a quaternary structure. The linear sequence of amino acids in Kras is connected through peptide bonds. The secondary structure is shown through folding proteins within the polypeptide chain, which is shown through coils. This over here is my Kras protein structure. So all of these coils represent the secondary structure. The 3D folding of the protein is shown through connections, which is through hydrophobic interactions and disulfide bonds. So all these connections over here and the overall way the structure is folded shows the 3D structure. So the key mutation in Kras protein is the G12C mutation, which is the glycine 12 cytosine mutation. This affects structure where the primary structure originally has glycine at position 12, which allows Kras to adopt multiple conformations. Cytosine is bulkier and has a reactive thiol group, which affects local interactions. The mutation introduces steric hindrance that slightly affects loop flexibility. So over here, this is a normal protein structure of Kras. However, this one location over here shows that it is cytosine instead of glycine. This represents the mutation within the Kras protein. So, how does the mutation affect the function? Overall, the thiol group of cytosine can form unexpected interactions with nearby residues. The altered flexibility of this loop makes Kras more prone to staying in the active GTP-bound state. It locks Kras in the active state because it resists GTP hydrolysis. This can lead to continuous signaling through the MAPK pathway, which promotes cancer because you're going to have continued cell division. So this is my protein model of Kras. So some of the limitations of my model is that, number one, it does not show the primary sequence of the protein. This model only shows the secondary and tertiary structure. Another limitation of my model is that the Kras undergoes conformational changes between the GTP-bound and the inactive GDP-bound state, but my model specifically only shows the active GTP-bound state. Lastly, another limitation my model has is that it does not show side chain interactions or orientations. This is a very general model of what Kras would look like. And this right here I want to point out again is the mutation over here where it is cytosine instead of glycine. This is just one position, and this one position can cause overall steric hindrance throughout the model. Thank you so much.